Hi, my name is Clarissa Brock. I will be presenting along with Jacquelyn, Morgan, John, and Kellen, and we are working on figure 3, E through H, in the primate Parkinson's disease model. So we're going to get into the research question and the goal of these figures and why we're doing this. So the goal is to help those who suffer from Parkinson's disease, which is a debilitating illness and characterized by tremors, postural problems, and rigid movement. It is extremely frustrating to live with, and scientists have targeted the specific neurons associated with it and are attempting different forms of treatments, which first began in rats and non-primate models and has now moved up the taxonomical hierarchy to primates, including monkeys. So what, with focus on the degeneration of dopaminergic neurons, which are correlated to the symptoms of PD, in the midbrain, scientists and researchers have used a number of modalities to understand as well as intervene with this disease and the deterioration in patients. We see TH, which is tyrosine hydroxylase, a catalyst for L-DOPA, and the transplanted human iPS cells, and focus on the striatum of the brain, which is a major function of movement for the body. So how in figure 3, E through H, scientists wanted to see, aside from the TH plus cells, what other significant findings could be found along with what was already discovered in these stained images. So next we will be looking at the approach taken, and Jacquelyn will be discussing that next.
So, as Clarissa mentioned just now, figures 3E through 3H, scientists are wanting to monitor traces of tyrosine hydroxylase in order to conclude the status of the grafted cells. So, the main experimental methods or approach used for these figures are dab staining or immunostaining. Now, you might be wondering, what is immunostaining? What is dab staining? How does it work? What does it do? How are these methods significant to not only this part of the study, but the study as a whole? Basically, immunostaining is a staining method that uses released antibodies, and these antibodies will detect certain antigens on the sample. So in this case, we are using dab staining, which is a type of immunostaining. And in dab staining, hydrogen peroxide will be oxidized, and this oxidation will cause a precipitation to form. So in order for this to happen, an enzyme is actually responsible for this oxidation. So the precipitate will form in areas where the enzyme is present on the sample. And after the staining is done, usually scientists will use light microscopy or a fluorescent light in order to view the sample, and the light will show what's visible on the sample. So here we are trying to find traces of tyrosine hydroxylase. However, there is a possibility to find other things. So we switch slides. You can see here, this is figure 3E, and TH stands for tyrosine hydroxylase. It's shown here in red. However, you can also see FOXA2. This is actually a protein coding gene that also showed up on the sample after the staining has been done. So FOXA2 is green and TH is red. I won't go into much detail about the significance of what this means, or, that will be done later when the results are explained. But yes, this is an example of what a stained sample would look like. And figures 3E, 3G, and 3H are stained images, or images of stained samples. So here, figure 3F is a bar graph. It's another method of collecting data. And basically, this is it's a comparison between the healthy primate group and the PD group. And it's comparing the numbers of TH cells and numbers of FOXA2 cells. So now I'm going to pass it on to Morgan, who will explain the importance of these results, what this means, the significance of these stained images.
So now let's talk about the results from the conducted experiment. As stated before, immunostaining is a process scientists use to detect a specific protein or proteins in a sample. In this case, FOXA2 and TH were sampled. FOXA2 is a protein that is a transcription factor that plays a significant role during neurological development when dysregulated or mutated. And tyrosine hydroxylase, or TH, is the enzyme responsible for catalyzing the conversion of amino acids. As seen in Figure 3e, the cells that survived showed a result of FOXA2 protein, and less than half of the monkeys showed presence of tyrosine hydroxylase. This is the same data as the previous slide, but represented in a bar graph. The x-axis, which is the independent variable, examined subjects 2, 4, 7, 11, 1, 6, 9, and 10's left and right brain cells. The dependent variable, or the y-axis, illustrates the number of TH and FOXA2 cells. There is no remarkable difference between the subjects that are healthy versus the subjects who have Parkinson's disease. Only 33.3 plus or minus 24.4% of all subjects express TH. However, this is not a factor that gave the scientists any more reason to research this further. The last two figures, 3G and 3H, represent markers DAT and GIRK2. This was used in comparison to TH in their merged images. The dopamine transporter, or DAT, is a membrane-spanning protein that pumps the neurotransmitter dopamine out of the separation of the two neurons back into cytosol. TH cells are present in figure 3G, which used with DAT. In figure 3H, GIRK2 is the potassium channel that regulates insulin by neurotransmitters. The pattern of both TH and GIRK2 are similar apart and when merged. Now John will explain how the research answer correlates back to the research question and provide a conclusion.
Hi, my name is John. Now I'll be discussing research answer and conclusion. So scientists performed this experiment in order to find traces of TH in the grafted cells, which would mean that the dopaminergic neurons are surviving. They performed a staining process called immunostaining and found TH and other marker genes with it. So the research answer is that TH was detected, and this shows that the grafted cells are surviving in the primates, making this a step further to concluding that using iPS derived dopaminergic neurons are working in the primates to treat Parkinson's disease. Further examination of TH cells through DAB staining revealed a correspondence between the marker genes obtained from the surviving grafted cells and the dopaminergic neurons acquired from the human iPS cells. Marker genes such as GIRK2 and DAT were found in the cells that survived after performing immunostaining. This comes to show that there's a relationship between the marker genes and the dopaminergic neurons that derives from the iPS cells. The results show that the maturation rate is low and is constant with prior observations of transplanted human iPS cell derived dopaminergic ancestors in 6-hydroxydopamine models with rats. The TH plus average numbers resulted in no difference between the grafts that came from the healthy individuals or the patients with Parkinson's disease. We can conclude that there was an increase in binding potential of PE21, and it was only observed in 2 out of the 6 monkeys. There was no serotonin found due to the use of choline, eliminating serotonergic neurons, and furthermore, the researchers found less than 1% GFAP or GABA cells, and there were no CHAT or VGL-UTI cells found. It was also concluded there were no certain pathological discoveries. Now Kellen will perform a professional critique and declare if the conclusion is supported.
So our group would support the conclusion that further examination of TH cells through DAP staining reveals a correspondence between the marker genes obtained from the surviving grafted cells and the dopaminergic neurons acquired from the human iPS cells. And so the evidence that brought us to this conclusion were from our results, and that the size and structure of TH cells similar to host dopaminergic neurons, and that those dopaminergic neurons are essential for the regulation of brain functions such as motor behaviors, and TH cells show similarities to the dopaminergic neurons in that sense. Also, that TH cells that survived the graft expressed FOX2A and TH proteins, and that the gene markers DAT and GIRK2 were expressed in the surviving TH cells. No pathological findings within the TH cells were observed when grafted. And so this data reveals that DAB staining of TH cells displays the survival of the dopaminergic neurons, and the grafting of the TH cells was safe through monitoring the cell survival. And so some follow-up experiment ideas would be to continue DAB stainings of TH cells to further analyze gene markers and their impact or correlation to L-DOPA treatment, as well as examine dopamine synthesis to further convey that the grafted cells function as dopaminergic neurons.